I don't wanna grow up, I'm a curious kid. The world has 100 questions I can play with. So I'll open my arms and eyes and wonder every day till the day I die. No one really knows why gravity exists.
Thank you all for being here. So let's get started. So as Tucker said, I'm a plant biologist, and that means that on occasions when I meet new people and if we are out in a park, hiking, maybe one of the beautiful parks in the East Bay, like Tilden Park, you're a plant biologist, so they'll point at whatever plants in the landscape and ask me, what's that plant? What does that tree do? Can you tell me something about this musk or whatever? And generally the answer is a bit of a peeved, I don't know, because although I'm a plant biologist, what I really specialize is in plant cell biology. And a cell biologist is very different from another biologist that would be studying taxonomy, for example, that would be able to tell you, an arborist that would be able to tell you what that tree is. What I study are cells. That's why I'm a cell biologist. And cells are the basic unit of all life on Earth. There is an organism that does not have cells, and having cells is a requirement to be alive. There are unicellular organisms, and unicellular organisms only need one cell to be alive. We'll hear from Rachel, I'm sure, about a lot of microbes that are unicellular. We, and pretty much all the organisms you may have been able to see with the naked eye, are multicellular organisms. That means, yes, we have cells, and we have trillions of them, and they're all organized into tissues, which then make up organs, and then a complex organism like our cells. And what I would like to sort of make an analogy of is a cell to a city. So if you see on the left, there is a plant cell. And the plant cell has a number of subcompartments in which functions are happening. The main function happening in the nucleus is the DNA, the genome is being protected. And then you have other compartments like the chloroplast where light is turned into energy, or the energy from light is turned into a usable form of the cell. And then we have a city where we have similarly subcompartmentalization. We have buildings like hospitals or schools or city halls, and they all serve their own discrete function, but they're all necessary for the city to make sense and for the city to function. And they're all connected through roads and other means. Of course, those were cartoons, so here are real pictures of both the city and the plant cell, and you can see those subcompartmentalizations coming to greater, you can see them better. You can see chloroplast, you can see other subcompartments, the nucleus that I was talking about, where the DNA is being stored and protected. But what you cannot see in the city or in any of these pictures, for example, in the city, you cannot see who the city was built for. Who was the city built for? Cities are built for people by people, and they serve people. What would be an analogy to people in a cell? Well, my argument is that the most important players in a cell that keep the city running, that build a cell and that keep it functioning and that keep it sense, are proteins. Here's one of my favorite proteins. It's one of the ones that I studied earlier in my PhD. Its whole function is that of pulling another protein that got stuck somewhere that it wasn't supposed to be stuck in. It's to pull it, physically pull it. We have models that show how this is done, and they pull it out of the situation where it's not supposed to be. Another example, and I think this is going to be very fascinating, this is really fascinating to me, is of this motor protein that you can see kind of walks over a pathway that is also built by other proteins. This is a motor protein that is carrying an important cargo vesicle, and the cargo could contain all number of important materials, and moves it across the cell through this walk-like motion. It's called a kinesin protein. So this could be your equivalent of a UPS driver or delivery person in a city. So proteins are fundamental to the functioning of cells, and they give every compartment of the cell an identity and a function. So let's take a look at how proteins are structured. Let's start from the most sophisticated level of organization that a protein is going to ever have, and that is called a quaternary structure, and I'm showing it here. A quaternary structure is a final structure in which the protein has sense. Here you see a crystal model of it, and what you can see is that there is a central pore, and that's where the protein in the previous slide was pulling another protein through, and it has it's composed of 6 smaller proteins that by themselves don't have any function, and they come together in this assembly that actually gets a function. And we call this a quaternary structure. If we go down one level, of less sophisticated but still sophisticated organization, we get the tertiary structure. And a tertiary structure is nothing more than the three-dimensional organization that the protein takes. It's the topology that it gets. Without the structure, it will not be able to do whatever function it is designed to do. It is not a two-dimensional molecule, it has a three-dimensional structure, and that's called a tertiary structure. If we go down one level of sophistication, we get to the secondary structure. The secondary structure of a protein is how that protein, which we'll see later as a protein chain, comes into two main types of organization, either an alpha helix, which is a coil, pretty much, and a beta sheet, and this is sort of almost like a fence-like structure. And here, from this previous slide, we can see multiple examples of alpha helices that are making up the lower portion of this protein, as well as beta sheets. I can see a couple there in the top part. So these are the ways that the protein folds to then take on a three-dimensional structure later on. But the most basic, the simplest level of organization that a protein can have is its primary structure. It is an amino acid chain. It is a concatenation of amino acids. Just like any chain that we're familiar with in the human world is made up of individual links. But unlike chains in our world, each link is different. It's composed of a separate type of amino acid. There are 20 naturally occurring amino acids, and each one is going to have its own typical and unique physical properties. It could be its length, it could be whether it's positive, negatively charged, or hydrophobic, meaning it has no charge at all. So this sequence, the identity of the protein, is determined by the sequence of amino acids that it contains, and that is going to determine later how it folds in all the three-dimensional structure that I mentioned earlier. So when somebody goes to the gym and works out, what they're doing is that they're intentionally breaking the muscle fibers in their muscles, and then they go and take a lot of different, I'm not sponsoring any of these, I'm just an example. They take a number of protein powders. Those protein powders contain amino acids that then will, hopefully, in the body, in the muscle cells, be turned into primary structure, into a protein that has a primary structure that then will fold into a secondary, later a tertiary, and then a quaternary structure. And then that quaternary structure will go into the muscle to repair the fibers that were broken during the workout, and hopefully make the muscle stronger, bigger, hopefully sexier, and that's how this is an example of how proteins are used in the body. So I hope I've driven away the point that proteins are the fundamental sort of workers and dwellers, citizens of the cities that we call cells. So then that begs the question, how are proteins themselves created? This actually takes us to the central dogma of biology. It's one of the most fundamental concepts in biology, and that is that all the information to make proteins, and the proteins which then make the cell and make everything work, all that information is stored in the DNA, the nucleic acid, the DNA. And that is our genome, and that is stored in the nucleus. It is the molecule that the cell protects as much as possible because it contains the vital information to make all the proteins that exist. And DNA is kind of like a big encyclopedia that contains all the information. Then for a specific protein to be made out of all that information, an excerpt of DNA is transcribed into a more compact form, a short form, and we call that messenger RNA. This is the process of transcription. The information is turned into RNA. That messenger RNA is then turned into protein in what will be the protagonist of the rest of the talk, and that's the process called translation. Translation because you're going from information that is stored in nucleic acid, one of the big main biomolecules, to information that is in a protein and then will fold and take on this function. This is a more granular vision of what I just described. The information is stored in ATGC sequences across the DNA. Then there are the relevant part to make a protein is transcribed into its own more compact form, into RNA, and then that RNA is translated into a protein that has the amino acid sequence that I was mentioning earlier. An example that we're now probably all familiar with is through mRNA vaccines. And what we know happened during the vaccination drive, and what the vaccines were all about, was inducing our bodies to produce the spike protein on the membrane, coating the surface of a SARS coronavirus viral particle. And how the vaccines work is that and messenger RNA was directly given to our bodies, and our bodies took up that messenger RNA if you got vaccinated, and translated that into the spike protein that then was recognized by the immune system, so that whenever a real viral particle would appear, then the immune system will be able to mount an immune response to the real virus. So you have the antigen, and the innovation here is that the antigen is produced directly by the human body when it's provided with the messenger RNA through the process of translation. How does that actually look like? So here we have sort of a cartoon animation of how that would happen. You have a molecule of messenger RNA that's coming from the nucleus where the DNA was stored, and that's where the messenger RNA was produced, and then it will float until it is identified by the new protagonist of our talk, the ribosome. It scans through this messenger RNA, it literally is reading through the sequence of the information that's there. And slowly but surely, a protein, an amino acid chain is emerging and folding into its three-dimensional structure. And eventually, it will detach and go on to do its function. So here I have just introduced one of the most important aspects of this talk, and that is the ribosome. The ribosome reads the messenger RNA, it turns the information that is in the language of nucleic acids and uses it to synthesize proteins. This is a 3D crystal structure of how messenger RNA would look like. And I call ribosomes the interpreters of life's code. And life's code in this case would be the information that was in the messenger RNA that came from the DNA. That's the code of life. But I have been intentionally provocative by calling these interpreters, because everything I've told you so far, and what you might have already perhaps encountered before, advanced biology classes, is that ribosomes are kind of like typing machines, very loyal, very well-oiled and well-functioning machines that simply take a message and turn it into, well, in this case, they translate it. Take a message that is in one language, turn it into another language. But there is no agency on the part of a typing machine to do anything with that message. It's just relayed, it's just turned into another form, in this case from messenger RNA to protein. But what I'm calling them is interpreters. Why would I call them that? Well, there is recent evidence, I'm talking about the past decade or so, that shows that they are more complex than just that. Here I am showing you two sets of ribosomes. One, these come from plants that have been treated with abnormally cold temperatures, it's a cold treatment, and a heat treatment, so abnormally high temperatures. And highlighted in yellow and purple, so in yellow are highlighted those components of a ribosome that are being downregulated, meaning that there is less abundance of that component of the ribosome. So these components are less abundant. And in purple, you see those components that are more abundant. And the picture is different, but with the same color coding for heat-treated plants. So here, there's actually a profound conclusion. The environment is changing the way that these supposedly very standard, very well-functioning, and very loyal machines look like. Their composition is changing based on the environment, which is not what you would expect of a machine that is supposed to just simply relay a message translated from one form to another. So there's something deeper going on. And actually what the community has been able to find and figure out over more years of more experimentation with a host of laboratory species is that instead of one type of ribosome across the cell and across cell types in the organism, just one simple machine, across a cell and across an organism, what you get is a heterogeneous population of ribosomes. Some ribosomes may have small molecules that are attached to them, making them slightly different than other ribosomes. Some ribosomes may have different versions called paralogs that are one part of the ribosome just switched with a similar but still different part. Maybe some factors, meaning other proteins, come in and enhance the ribosome. Maybe some parts drop out. And maybe some of the components are more or less concentrated, which is kind of the case of what I was showing you earlier. Some of the components go down in concentration, some of the components go up in concentration. This means that all the ribosomes in a cell are not alike. They're not alike across the organism, and this is called the heterogeneous ribosome hypothesis. If we can more robustly provide more evidence for this hypothesis, it would redefine the way that we understand proteins to be made. We understand them to be just simple product of messenger RNA being translated, but what we have actually been able to sort of infer through experimentation is that ribosomes get to pick which RNAs to turn into protein at which times. They seem to have through some unknown regulatory process, the ability to pick which proteins to make at which points of the cell's life or depending on the environment. So this is completely different than just the function of turning a messenger RNA into protein blindly. There's agency on the part of the ribosome to decide which proteins get to be made. And getting into why this is relevant for us as humans, if we can understand, if we can corroborate these findings and get this new understanding of how proteins are made, we can then go into all sorts of applications, both biomedical and biotechnological. For example, if we go back to the messenger RNA vaccine case study, if we know that there are some RNAs that look better to the ribosome, then we can, when we create these messenger RNA vaccines, we may be able to make messenger RNAs that are turned into protein better, that the ribosomes like more. And therefore, when we give these messenger RNA vaccines to patients, they may be able to have a better immune response, produce more antigens and then produce a better immune activation. We have already actually observed that some diseases like cancer and neurodegenerative diseases, neurons in patients with neurodegenerative diseases have ribosomes that are specifically producing the proteins that are causing those diseases. So if we can understand this better and we can target those ribosomes independently, we may be able to have another tool to attack these diseases and cure people. Finally, going back to the plant sciences, which is sort of my neck of the woods, if we understand which proteins, which ribosomes are producing which proteins preferentially, we may be able to then breed and engineer plants that can produce more of the proteins that make grain, that give grain their nutritional value, that make plants resistant to pathogens, to stresses, to pests, and that perhaps are more fit for the changing environment that our sort of planet is going to face in the coming years. So in summary, and to finalize my talk, if I would sort of see into the future, I hope that we can correct our understanding of how proteins are created and use this newfound knowledge through biomedicine and biotechnology to improve all of our lives. Thank you. And I'll take any questions.
So I'm wondering about the aging process and ribosomes. Being able to turn on and turn off the production of proteins at different parts of the life. I understand there's maybe some other, I'm just wondering how much are ribosomes being looked at in that regard?
So ribosomes are definitely being looked at now that we understand that there's a difference in ribosomes across cells and across the organism. They're being looked at in every single disease, and aging can be treated sort of as a disease in a certain way. And so we can look at whether those ribosomes are not functioning properly as the body ages, meaning that they're not as able to produce some of the proteins that in younger individuals may be able to perhaps deal with some of the disruptions that eventually lead into aging. So ribosomes are being looked at through these lenses. They're being looked at through all lenses of medicine. So how do the ribosomes in a certain diseased tissue look like? And how do they degenerate throughout disease? And aging can be considered as a disease. So yes, they're being observed through those lenses as well. How do the ribosomes change? Do they not perform so well during aging? And is there any way to induce the body to make it perform better?
Just wondering, when this heterogeneous ribosome hypothesis, I'm wondering what year when was that first proposed?
Yes, so it started getting really mentioned as a hypothesis on its own, I would say in the 2010s, early 2010s. There was already evidence from decades prior that there are multiple versions of, not ribosomes, but the components of the ribosome. It looks like, okay, to make that one part of the ribosome, there are multiple genes that do that. And so there was some debate as to what that meant, and then people observed that some particular mutations in some of those genes, but not others, led to some type of consequence for the cells that the other mutations didn't. That was in decades past, early 90s, and sometimes in the 70s, but it's really by the early 2010s that this was kind of coined as a heterogeneous ribosome hypothesis, that there is actually a functional role to different ribosomes, that this difference actually matters to something. Yeah.
Also, I'm wondering about ribosomes in humans versus plants.
Yeah. So ribosomes are, because of how critical they are in the synthesis of proteins, they are very well conserved across all organisms. There are ribosome, and by ribosomes here I'm sort of being very general. Ribosomes are made themselves of proteins. So ribosomal genes and the proteins that come out of those genes are very well conserved across life. There are universal particles, there are universal components, meaning they're shared across all known species. There are some that are only shared with eukaryotes, which just means cells that have a nuclei, so not bacteria, not some microbes. So there is a lot of structural similarity to the way they look, but where the differences start to play in is in this heterogeneity question. It's possible that plants modify their ribosomes after they're created in ways that animals do not, and vice versa. And there are ways in which there could be a preference in the type of ribosome or ribosome state that exists in a plant cell that is not preferred by an animal cell. And this is always going to be tissue dependent, right? So ribosomes in one part of the body may not look similar. And because plants have completely different organs, then this would be even more so the case. Yeah.
You said something about how the cell protects the DNA within the nucleus because it wants to protect it. But
yes.
Obviously cells don't have brains and logic and free will and stuff like that. So it must come down to some sort of physical structure that forms. Do we understand what it is down at the physical level that makes proteins and other building blocks connect in the way that they do, so that they're consistent from one cell to another?
Yeah, so if I understand the question correctly, it's what do we understand at a, I guess, molecular level, what is happening that is, well, in the case of you mentioned, protect DNA, but also make proteins take on the shape that they do. We understand it up to a certain point. So right now, with our current abilities, we are able, there are some artificial intelligent models that can predict the way that a protein is going to look like in its three-dimensional shape, just based on the physical properties that it has. You know that it has a set number of amino acids that are all positively charged or negatively charged, and so you can predict how it's going to end up. And those models work up to a certain point. There are a lot of limitations because cells end up doing a lot of post-translational, I guess, post-production changes that are, we know how they happen. For some of them, we know what the physical process is, but it's always hard. One of the biggest limitations in biology is that it's hard to follow something across the entire life of the cell and to know exactly what happens to a protein across its entire lifespan. It's something that takes a lot of studying. But yeah, we understand the principle of why proteins take on the shape that they do. And we also have seen that there are proteins that simply refuse to actually have a shape at all, and they are intrinsically disordered, and that is another entire field of biology that deserves attention, is why some proteins just do not fold at all, and that actually has some advantages. That is really getting into biophysics, and it's also beyond my expertise. Does this answer your question?
Yeah.
In about the middle of your talk, I think you snuck in the word agency, saying that ribosomes or whatever particle you were discussing don't have it. But then you showed us the videos that, I don't know about agency, but it's so incredibly complicated. I want to understand where agency would appear at that micro level. Does it ever?
Yeah, so I wanna be responsible scientist here and say that I use these terms to help people understand what I mean. I do not actually imply that there is real agency as we humans understand it. It's a construct. I'm using those words to help sort of the message get across. What I perhaps should have said is the ability to discriminate based on the properties of, for example, messenger RNAs. This hypothesis of the heterogeneous ribosome postulates that the physical properties of some ribosomes, just by the nature of how they are, sort of do not fit very well with the way that some messenger RNAs that produce some proteins are. And maybe to get to your bigger point of what explains this immense sophistication, that is actually more of a philosophical question. This sophistication started, we believe, millions of years ago with the evolution of the first living organisms, and it's just one figment of probability. Of all the possible things that a soup of inert material could produce, the right combination of chemical reactions led to the first life form. And that first life form probably died off immediately, but this happened over until complexity sort of naturally built on top of itself, leading to some of these self-perpetuating chemical reactions. At the end, life, if you want to look at it from that point of view of biochemistry, it's a self-perpetuating chemical reaction of all the possible outcomes that would lead to just a massive goo that doesn't move. On Earth, with the right conditions, the right amount of time, we got a chemical reaction that is so complex that continues to go on and on and on and on. And then develops into this very sophisticated, almost intelligent-like organism. And we are intelligent ourselves, of course.
Of course. All right. Hey, Enrico, thank you very much. Our second speaker this evening is Rachel Porter. She's a researcher and PhD candidate in biophysics, a topic that was just referred to by Enrico, so maybe she can help us here, in biophysics at Stanford. Rachel grew up in, and I quote her, very rural Texas, and earned her bachelor's degree in molecular and cell biology at Texas A&M University. Before coming to Stanford for her graduate work, Rachel lived in Slovenia, studying extremophiles, critters that survive and perhaps even thrive under extreme environments, as a Fulbright researcher. Please welcome biophysicist, rock climber, and Wonderfest Science Envoy, the former Texan, Rachel Porter.
Well, thank you all for coming out here tonight, and thank you for the introduction, Tucker. As Tucker mentioned, my name is Rachel. I'm a biophysics PhD student, and I work in the lab at Stanford, this lab of Dr. Casey Hall. So in this lab, we study the human microbiome. My particular project is investigating the spatial structure of the microbiome. So a lot of people are interested in what microbes make up this group of bacteria that live inside the human intestine and help us digest food. And while that's very interesting, I'm not only interested in what bacteria are there, but how they organize themselves. And so today I'm going to try to kind of use Enrico's analogy and compare bacterial colonies to cities. And I hope I can convince you by the end of this talk that they are very competent city planners. And in fact, bacteria have their own version of zoning laws. They have transport infrastructure, and they also undergo gentrification. So by the end of this, I hope you can appreciate that these are not just simple little unicellular bugs floating around, but they are very complicated. But before diving into all of that, I want to orient you to the system that we work in when we study the human gut microbiome. So as I mentioned, this is the group of trillions and trillions of bacteria that live inside the human gut. So we're looking at a rough outline of the human digestive system here. And so we start at the stomach, And food then winds through the small intestine before making a pass through the large intestine and then exiting the body. So during the process of digestion, most of the breaking down of food happens in the stomach, and the bulk of the absorption, particularly of small simple sugars and simple fats and proteins, will happen in the small intestine. Then it's just kind of the nasty, hard to digest bits of fibers and other kind of tricky chemicals that will make it into the large intestine. And that is where the microbial magic happens. So what I'm showing here is a snapshot from a study where we looked at some of the bacteria that were found in the large intestine. And then these were treated with different dyes and stains so that different kinds of bacteria appear differently. And what you can see by looking at this is that not only are they incredibly dense, they're also highly diverse. So now I've explained a system where we have 3 different major organs, and then even within these organs, we have different kinds of tissue types. So for example, in the large intestine, there's the center region where food and poo will pass through, and around that, there are these kind of finger-like appendages that we call microbiology. And these serve to increase the surface area of the large intestine so that it can absorb nutrients more efficiently. And coating these microbiology is a kind of layer of this jelly substance called a mucus, or mucosa. And this kind of covers the cells and makes sure they don't come in direct contact with any harmful bacteria. So even within a single organ in the system, there are many different places that bacteria can dwell. So now we have 3 organs, many tissues within these, and oodles of kinds of bacteria. So I hope you can appreciate this is a very complicated system. And the questions I'm interested in answering have to do with very fine details of bacterial organization, which are almost impossible to get at in the system. So in order to answer these questions, I primarily work with a simpler model system, which is bacterial colonies. So what we're looking at here is much simpler. I'm showing a little petri dish that has basically the equivalent of bacterial jello growing on it. That's the kind of clearish light yellow stuff. And smattered across the surface that look like light little spots, these are bacterial colonies. So you're probably looking at them and thinking, wow, how are you going to spend 20 minutes talking about these? These are not exciting at all. And so I hope by the end of this I can convince you that yes, they are extraordinarily exciting. So what looks like a teeny tiny speck is actually billions and billions of cells. And they're not just in simple little piles like we used to think. They're highly organized. So I'm going to give you 3 examples throughout this talk to prove to you that these are very, very organized colonies. And so throughout this talk, I'm going to use 2 different kinds of schematics to demonstrate some of the things these colonies are up to. And so the first one here on the left is going to be, if you can imagine, we have this petri dish, we cut it right down the middle, and then we're looking at the side of a cross-section of a colony. So this sort of yellow region here is the bacterial jello that has all kinds of nutrients and good stuff that lets the bacteria grow. And then this white region right here is the dense bacterial colony. And then above that would be the air. In some other schematics, we'll be looking at a colony from the top down. And so that's what we see here. The little circle is the colony, and the surrounding area is going to be the nutrient jello. So, without further ado, let's get to the city planning. So first I'm going to talk about zoning laws. And so what we're looking at here is a map of San Francisco. And shown in pink are single-family residence zoned areas. There are other residential areas shown in blue, and then non-residential areas are shown in gray. So we can see, Golden Gate Park, and then a bunch of commercial districts. And the overall goal of this is to kind of keep the city organized and help it function smoothly, by instead of having everything happening everywhere, have defined zones where different processes happen. In the same way that we do this with cities, bacteria do this in their colonies. So what I'm showing here is a schematic of a bacterial colony color-coded based on the type of metabolism it uses. And so how does this happen? In order to explain that, I'm going to have to give you guys a crash course in metabolism, so bear with me. And imagine that you are a single bacteria growing on this nutrient jello. The first question you're going to ask when you're thinking about how you can get energy is probably going to be, what are the nutrient sources available? So, one common option is going to be sugar. The particular sugar I'm thinking about today is glucose, which is very common for bacteria. It's common for us too. You've probably gone to the doctor and they tested your blood glucose level. It's a very easy form of energy that bacteria and humans use, but I'll just refer to it as sugar. So, say we have this sugar. The next question we'll ask is there oxygen? And if the answer is yes, That's great. We can use this process called sugar respiration, where we start with sugar, we go through a bunch of chemical reactions, and then at the end, we produce carbon dioxide. And throughout that process, we get a lot of energy out. So this is a really good, highly efficient way to get energy. The kicker is you really have to have oxygen. If you don't have oxygen, that's okay. You can use another pathway called sugar fermentation. And so this is basically the first half of sugar respiration. It's just once you get to the steps where you need the oxygen, you have to quit. So we start with sugar, we go partway through, we develop an acid, and then we just let the acid go. So these are two different ways that bacteria can get energy. And in case this sounds familiar at all, these are also ways that humans get energy from molecules as well. So most of the time we undergo this kind of sugar respiration. We breathe in oxygen, we eat sugar or foods that can be converted into sugar, and we exhale carbon dioxide. Sometimes, however, we use the sugar fermentation. So for example, if you've gone to the gym and felt sore afterwards ever, that's because some of your muscles weren't getting enough oxygen. And so they're forced to switch to the sugar fermentation pathway, which then causes an acid called lactic acid to build up in your muscles, and that's what causes the sensation of being sore. So both humans and bacteria do this. Now, if we're a bacteria and we don't have sugar, then we're going to have to look for other nutrient sources. And another common one will be acid, particularly the acid that is produced from the sugar fermentation process, which is called acetate, but I'll just call it acid for now. So let's say we have this acid. The next question will be, again, is there oxygen? And if there is, great. We can basically pick up where sugar fermentation ended, and then take this acid and process it all the way to make carbon dioxide. So that's great. We can still get energy and grow the cells. And then if we do have acid but we don't have oxygen, we're kind of out of luck. So this is all the metabolism you need to know to understand how colonies have these zoning laws. Let's see how they play out in the context of a colony. So we are looking at a bacterial colony, and we've got air above, which is rich in oxygen. We have this nutrient substance, the jello below, which has plenty of sugar and plenty of oxygen at the beginning. And then we have our colony here. So early on, all of the cells kind of along the edge here that are in contact with that jello are able to get sugar that kind of diffuses up from the food, the nutrient jello. And that allows these cells to undergo sugar respiration because they have both sugar and oxygen coming from that jello. However, after a while, the cells start using up all of the oxygen. And so it's harder for the cells in the middle to get some. So this means that the cells have to switch to a different kind of metabolism. They switch to sugar fermentation. So as you'll see here, at the very edges, cells continue using the sugar respiration because they're able to get oxygen from the air around. But cells that can't get oxygen from the air have to switch to fermentation. And this produces a byproduct of a lot of that acid. And so that acid will accumulate and actually float up into the upper regions of the colony. If you notice, these are also going to have contact with the air above. So these are perfectly primed to undergo acid respiration. And so through these kind of simple rules, cells are able to organize their metabolism, just like we organize our cities. So on to example number 2, which will be transport infrastructure. So now I'm showing a map of San Francisco, some of its major thoroughfares. Of course, there are plenty of other streets that go through the city, but if you're trying to get from side to side, you don't care about those so much. You want to think about the main roads that'll get you there fastest. And so these kind of help us get things from place to place efficiently. This is something that bacteria also need to do in their colonies. And so to meet that end, they've come up with a similar solution, which are these sort of tunnels that function like highways. So for this example, we'll be looking at colonies mostly from the top down. So as this colony first grows, it's able to get most of the nutrients it needs from the jello right underneath where it's growing. However, after a while, it starts depleting that, and it needs to get energy or energetic molecules from the zone around the colony. Once this happens, it kind of has a problem because that is a really, really tricky thing to do. And the reason for that is because nutrients move through a process known as diffusion. So a good example of the process of diffusion is if someone was to stand in that corner and spray little spurts of perfume. Immediately, you would be able to smell it really well right there. But in the back corner of the room, no one would be able to smell it. However, after a little bit of time, even if there's no wind or air movement in the room, you would be able to smell it faintly at all positions in the room because it will naturally move from areas of high concentration to areas of low concentration. So this is kind of the process of diffusion. If we want to think about it in terms of a city, you can imagine the process of a nutrient molecule diffusing from the outside to the inside being kind of like if you had to drive from the outskirts of San Francisco right into the middle, but you had no map, and every time you hit a little intersection, you just had to take a random turn. It would probably take you a very long time to get to the middle if you ever made it. So this is not an efficient way for nutrients to make it into the cell. In fact, if this cell is about 2 millimeters in diameter or so, it would take an hour by diffusion for nutrients to get from one side to the other, which might seem kind of fast, but these cells can divide once every 30 minutes if they have enough nutrients. So if you're waiting to divide, that's a pretty long time to wait for food. So, what do cells do? They build highways. So different bacteria do this in different ways. There's one bacteria in particular that does really beautiful systems. This is called Bacillus subtilis. So I'm showing a pretty old colony of this bacteria up here. And as you can see, there are some ridges that connect the nutrient-starved center to the nutrient-rich outside of the colony. And if we're to look at one of these ridges up close, we'll see that it actually looks like a little tunnel that these bacteria have built. And so the scale bar on there is 100 micrometers, which is probably the width of about 2 human hairs. So for us, this is very, very small. But for bacteria, this is huge. One bacterial cell is typically about 1 micrometer long, so that means 100 bacteria will fit in the length of this. So it's a huge architectural undertaking for these cells to make this. But they do it because it's so important. These channels allow nutrients to freely flow into the center of the colony and also allow waste to exit. And so, just as we have developed highways to kind of help transport things in and out of cities, bacterial colonies have developed these kind of highways, or perhaps a better analogy is tunnels. So, on to the final example, gentrification. So here's a map of actively gentrifying areas in San Francisco. And so gentrification is a process where an established community starts being kind of kicked out of an area as active growth happens. So we typically think of neighborhoods that are well settled and haven't been changing much for a long time, that all of a sudden have people coming in and changing the area, kind of driving up rent and making the people who have lived there for a while go somewhere else. So just as this happens in cities like San Francisco, it happens in bacterial colonies. So what I'm showing here is a cross-section of a colony that we're looking down on from the top. And this colony was taken, or was treated with a lot of different dyes and stains, so that slightly different types of cells have different colors. And what you can see here is there are a ton of little specks that look different from the background. So what I haven't told you about bacterial colonies is that most of the time when we grow them in lab, we start with a single cell on a petri dish. Then that cell grows, it divides into 2, then into 4, 8, 16, 32, and so on until you have billions and billions of these cells. And so, because they all came from the same starting cell, we expect these to be genetically identical. But what we see here is they're not. So how does this happen? So, again, we will be thinking about this looking down at a colony from the top. Historically, we thought that growth only happened in aging colonies kind of around the edge here. This is where cells have the easiest access to nutrients, and once a colony kind of has reached a couple days of age, it doesn't grow that much anymore beyond these regions. As we've recently found out, however, there are lots of little spots in the middle where growth will start happening again. This is what we see happening in this image. And to understand how this is actually occurring, we're going to need to zoom in and think about things at the cellular level. So, what I'm showing here is kind of a dense cluster of cells that would come from kind of an older region of the colony that's not growing so much anymore. So imagine, these cells are sitting here. They're in what we call stationary phase, where they're not really growing or dividing, they're just kind of trying to hang out as long as they can. And even though they're not doing much, they're still susceptible to mutations. So mutation is anything that changes the permanent code in the DNA. This is something, humans think a lot about when we go outside because UV radiation will cause mutations in our skin cells. And so, we try to avoid that to protect ourselves from cancer. These cells also have a lot of, dangerous mutating agents in their environment. And so eventually, some of them will get mutations. And so tons of different mutations happen in these aging colonies, and they have lots of different effects. But I'm going to focus on one kind that happens pretty regularly in these colonies. And this causes those mutant cells to secrete a chemical known as indole. So this chemical will go exit the cell and enter neighboring cells. And once it's there, it's actually lethal to them and will kill these cells. Once these cells have died, they'll kind of start disintegrating and all their contents will leak into the environment. And actually, fortunately for the mutant cell, these can be used as a nutrient source. And so this enables this cell to kind of break out of the stationary phase it was in, where it wasn't growing because there weren't any nutrients, and now it's found a way to convert its neighbors into those nutrients. And this allows the cell to kind of grow and establish itself and continue this cycle at the expense of the cells that were already there. And this results in patterns that look like this, where we see novel mutants kind of budding in a uniform background. And this looks a lot like gentrification. And so, I hope I have convinced you today that bacterial colonies are incredibly complicated. As we've seen, they have zoning laws, they have highways, they even undergo gentrification. So now with this increased appreciation of just how complicated colonies are, we're even more excited than ever to see what's happening, not when we're looking at a simple environment such as a petri dish, but once we're looking at an incredibly complicated environment of the human gut. So with that, I will thank you for your attention, recommend a couple reads for more information on the microbiome, and take any questions.
The tunnels that you talked about,
Yes.
what are they made of?
So interestingly enough, the upper region is actually made of cells. The center is hollow. And so it's a really cool process for how these form. Basically, it starts with a colony that's flat, and during the early stages of growth, there will be a region where cells are super dense, and they eat up all the nutrients so fast that this kind of stripe or spot will have a lot of cell death. And the dead cells just don't stick that well to the bottom, so then as kind of pressure builds in the surface, it'll pop up and form the tunnels.
Oh, so they don't like building,
Well, they grow and make it.
they grow it.
But they build it by growing and then it's shaped into the tunnel.
You have matched all the descriptions that I have been thinking of, in terms of cell, bacterial structures and all that. I'm an urban planner, so I can definitely relate to your entire, presentation, which is why I am hell yeah, this is definitely, my expertise, and, I commend you for, making those connections.
Thank you so much. I'm glad my city planning knowledge was sufficient.
No, seriously, it's the only thing that's missing now is, if you have, other things, you have the good bacteria, but you also have to deal with bad bacteria, Salmonella, E. coli, and so on.
Mhm.
If we could have a little extension of that one, then we could be dealing with, crime statistics and, all sorts of population data. No, seriously, it's one of the more profound, comparisons that I have ever seen that, as an urban planner here, I commend you for that excellent presentation.
Thank you. And I will say that multi-species colonies is kind of a growing area of interest. So maybe someday soon we'll have the kind of bacterial demographic information.
So on your gentrification part of the metaphor, what happens though when you have too many of those cells that are eating up the ones around them?
That's a
Is
good
that
question.
kind of, it sounds dangerous.
Probably. That's really dangerous for someone. So I think looking at this picture, we can kind of see that there are regions where it looks like some of these are overlapping, where we have some little purple ones and red ones. So I guess at that point, two different mutants that are really good at taking over areas have come into contact. Someone things out. So it is kind of interesting to think about this in the long term, because a lot of the mutations that allow cells to start killing their neighbors are not really good for the species in the long term, because they just start killing their neighbors, which is not great. So people have kind of hypothesized that maybe this is a very desperate measure just to keep some cells in the middle alive, and then maybe later they'll be the colony will be able to get some samples somewhere else and take over a new area. It's not really clear at this point in time, because it's hard to imagine this would be beneficial in the long run. but many different species do it. So we're still trying to figure that out.
You had 2 books up there.
Yes.
On the screen. The one by Ed Young I read, and it's marvelous. But yeah, lots of information, really. What about the other one? Why are you recommending The Good Gut?
So I will be honest, The Good Gut is the one that is on my reading list, and I haven't gotten to it yet, but I've heard incredible things by some of my lab mates who also work in the microbiome. And it was actually written by a professor, Dr. Justin Sonnenberg, that my lab does close work with. And so his work focuses a lot on not so much like the fundamental bacterial side of things, but more on like, this is what your diet does to affect, how you feel and how you live and how your microbiome looks. So it's really kind of medically oriented. And I forget the name of it, but he was also recently interviewed on, I think it's called You Are What You Eat. It's a Netflix series. So he's kind of an expert in that field.
Thank you.
Mm-hmm.
I'm an Ed Yong fan, and I have not read this, so thank you for the recommendation. Can you give us just a tantalizing hint as to what he means by "and a grander view of life"?
So I can give you my spin on the grander view of life. And so one super fun fact I did not share with you in this presentation is that if we just count the number of cells in the human body, 50% of them are human. 50% of them are not. And that's kind of a scary statistic. If we're just thinking about the number of cells, we're basically half human, half bacterial. And actually, if we think about the number of unique genes in the human body, because there's so many different bacterial species, like 99% of our genes are bacterial, and like 1% might be human. So most of the things that happen in our body by just kind of the number of unique events is probably going to be dominated by bacteria. So I think this is kind of a challenge to the human-centric view of humans.
Yeah, thank you.
Yeah. Yes?
How big can the bacteria colonies get?
Ooh, so there are actually some experiments where people will make, to try to figure this out, huge petri dishes, like some that are, bigger than this table. And if they have enough food, they can just keep growing for a very long time. Of course, like if, we start with the bacterial colony here, and it grows for a couple weeks and gets over here, the initial cells are probably not doing so well, but it can cover a really massive area. I don't know what the biggest bacterial colony ever is, but I do know fungi kind of behave similarly, and they can go underground for miles. So, pretty big. Yes?
So, I hope this isn't too ignorant of a question, but I remember, like, when we eat something, I thought there's some acids that come into play to decompose, and just one of my first question would be, I'm just curious to know how they're supposed to be pretty strong, and I'm wondering how that plays with the bacteria, and then also yogurt is supposed to be something very healthy, and so that's kind of a second. I'm just wondering, as yogurt has bacteria in it,
Yeah.
I'm just curious about the acid. We talk about stomach acids, and yet, in ways that, anyway, stuff.
Yeah, absolutely.
Not, really like caustic acids.
Right. The stomach is like a horrible environment if you're a bacteria. I mean, I think if you're anything, that's horrible. And so part of this, of course, is because we do eat food that's like coated in bacteria. Some is good, some is bad. And the stomach's job is not only to break up our food, but to try to prevent any like harmful bacteria from making it into our gut. So I would say, from what we know so far, is that most of the human microbiome is kind of established within the first 3 months of life. After that, it's a lot harder for things to get in there and actually stay in the intestines. So what I would imagine, and I'm not 100% sure, but I think is during this infant development phase, the stomach might not be quite as caustic yet, and that allows some bacteria to make it through and colonize. However, in later life, we do know that it's very hard for bacteria to make it through and actually colonize the human gut. As a matter of fact, even if the bacteria make it through the stomach, we found that taking some new invader bacteria and mixing it with a culture of an established gut microbiome will actually just kind of kill off the new member, and the culture will stay the way it was before. So it's very hard for new species to invade. And this is great because like if we add salmonella into a healthy gut, salmonella probably doesn't have much of a chance, which is what we want. However, if you have kind of a sickly microbiome, then salmonella can actually invade, and that's where it gets dangerous. As for yogurt, unfortunately, the same thing is kind of true with probiotics, where we can add all kinds of fun, juicy, bacteria into yogurt, and it seems like very few, if any, will even make it into the gut. And of those that do, the cells that are already there are going to compete and kind of kill them and kick them out. So if you have a healthy microbiome, that's what happens. Of course, if you're missing a lot of key microbes in your intestines, then maybe they stand more of a chance of settling in. But this is kind of a big takeaway I've gotten from Justin Sonnenberg's work, who's the author of The Good Gut, is that a lot of times you think about probiotics as very healthy, and it's true, but it's not because of the microbes, it's kind of because of what the microbes do to the food before we eat it. So their impacts on what's already in yogurt is more important than the fact that the bacteria are there, is what some of his research suggests.
You have time.
Yes.
I hear a lot about the vagus nerve, and I don't know if that is in the purview of what you're talking about, but how we think everything comes from the brain, and there's a lot of talk about how the gut it's the direct communication, right? And so how the vagus nerve communicates to the brain sometimes, and it's the gut that's a little more in control sometimes.
Yes.
I don't know. What
So this is definitely a region that I'm not an expert in, but I've heard this referred to a lot as the gut-brain axis. And so another kind of interesting piece of evidence we found that supports this idea that the gut has a huge control over how we think and how we feel is that gut microbes make a lot of serotonin. And serotonin is a hormone that kind of is related to a lot of like human emotions. And some of it's coming from our microbes, it seems. So I think there's a lot more research that needs to be done to prove that the serotonin produced in the gut actually makes it to our brains and is signaling there. But it's certainly interesting. I think kind of supports like we might have feelings, and it's because our microbes are feeling something.
Yeah.
Just kind of on that, I read a bunch of stuff and heard a bunch of stuff. Who knows if it's hooey or true, but people who eat so much processed sugar and so much bread and sugar and pastries and all of these things, they're more likely to have dementia because their bodies just aren't processing stuff right. Is there any of that?
Yeah, so I think the kind of issue with a lot of like kind of processed foods and foods that break down into really simple components, like, pure sugar or like really simple carbohydrates, is that basically that stuff just kind of gets dissolved in the small intestine, uptaken right there. And so there's not a whole lot in the diet then that will go into the large intestine, feed these microbes, and then contribute to any signaling that they're doing to promote health throughout the body. So I don't know the particular research connecting with dementia, but I absolutely believe there could be a connection. And another dangerous thing about not eating some like good fibery foods is sometimes, if you don't eat a lot of fiber, there's not a lot to feed your bacteria, and they can actually start to eat the mucosal lining of your intestine. So you want to keep them happy, give them fiber.
Oatmeal?
Yeah, oatmeal.
My husband and I used to travel in Thailand a lot, and we would go to the Chao Phraya River, and there were houses for people to live in that were on stilts above the river, and you go along and you see people peeing in it, dead animals get thrown in it, garbage gets thrown in it, the wash is done in it, everything is done in it. And I wondered, how can they survive? But I was told that the first thing they give to a child, before even breast milk, is water from the Chao Phraya. So now that makes some sense.
They've got some built-in immunity.
Right.
Wow.
All right, thank you. We'll have a round of applause for Rachel.